That was hilarious.
Thanks. I consider myself to be like in the 90th percentile in humor. You know, top 10%.
But everyone in this class will be like that.
Yeah, listen.
So the quiz that's due after class today
Yeah.
is based off class now? Or
Yes.
Class time?
No, class now plus what the lecture I posted yesterday morning.
Okay.
Yeah.
But last class did nothing.
No. All right. First things first. Were there any questions that came up? I don't know. I still didn't delete this last one. It was left over from the previous. I don't know. I left it there. Yeah,
Can you
gotcha.
go over the microarray?
Yeah, microarray. Yeah, and so a microarray is basically you just use a solid surface and you pack that solid surface with all of these different DNA fragments. And it is possible now to actually put a DNA fragment on this surface for every single known human gene. You can have a DNA fragment that will bind, that is complementary to the mRNA of every single human gene. And then you could take a sample from a particular tissue type in that person. Now this would be a very expensive microarray, you know, if it's gonna have every single human gene on there, and it's gonna be big. But anyways, and then you can take a tissue sample and you'll expose the microarray to the tissue sample, and if there is any of that mRNA present, it will bind to that DNA marker, that tag, or what do we call them? Tag's not the right word. Probe. Yeah, that's the right word. To a specific DNA probe that again it's complementary to a very specific RNA, or you can have it be complementary to single-stranded cDNA that you can induce to form by putting your tissue sample into a solution with reverse transcriptase, so that it gets written back into DNA but single-stranded. And again, you expose it to the surface that has probes that are complementary to whatever your target genes are. Like, so let's say you have a question. Let's say you have a question and you're like, okay, we have a suspicion that this specific gene correlates with this specific genetic illness. And so what you do is you take two people, one without the genetic illness, one with the genetic illness, and you take tissue that you know is impacted by that genetic illness, right? And you expose those tissue samples, one from the infected person, one from the uninfected person, unexpected person, uninfected person, and you put them both on this microarray or chip that you might call it, that has probes for a very specific gene, right? Say you're only concerned with a very specific gene, and you have a suspicion that the person that's infected with this genetic illness is expressing that gene, the person that is uninfected is not. And so you expose both their tissue samples to this chip with the probes that target the mRNA if that gene is being expressed, and then if they bind, it shows up. It basically, there are different styles of microarrays. Some of them will fluoresce when a probe binds. Some of them will just change a little bit when a probe binds, or you can go in and then wash it away and wash all the probes that aren't bound off, and then the only thing that remains are probes that actually bound a piece of RNA or DNA. Does that help? Yeah. Okay. So it's very simple. It's very expensive. So in the video I tried to make it clear that the value of microarrays is that they're very late, or let's just say this, they're not very labor intensive. They're very simple. Okay, they're very simple. You isolate some solution. You order this chip or this microarray from a company. They put the probes on for you, and then you just expose it to your tissue sample. Other things might be more accurate. They might be better, like RNA sequencing might be more accurate and it has more functionality, but it's a lot more labor intensive. It's a lot more labor intensive. And labor intensive typically means expensive, because the most expensive part in some of these things, not in all of them, because some of them, the materials are really expensive, but a lot of times the most expensive part are man hours. And RNA sequencing has a lot of man hours at the back end when you're trying to take your sequences and analyze them. Your microarrays, it's very simple. You take your microarray and it's like if the probe is bound to something, that gene was being expressed. It's very simple. You knowing what you know now, I could hand you a chip and you could tell me whether or not the target gene was being expressed by looking at this microarray. And you have zero experience with microarrays, I'm assuming. Do you have any experience with microarrays?
Not doing this.
Yeah. And you could do that. But if I gave you an RNA sequence and said, what does this mean? Man, that would be tough. That would be tough. Yeah, listen.
But isn't it labor intensive like on the front end because someone has to make the microchip and label it and stuff?
Yeah, but a company will do that and mail it to you. And that's actually fairly simple. It's fairly simple and fairly easy now to design a specific piece of DNA. Now, to know what your target probes are, yes, that's labor intensive. But you have to do that for RNA sequencing as well.
But right, so you have to do that for RNA sequencing, just not the person that orders this chip. So it's still the same amount of work, just different people doing it.
Yeah, it's not the same amount of work. No, you can make a chip for a microarray much easier than you could isolate a target RNA, eliminate all other types of RNA, and get it ready for sequencing. Yeah. Excellent questions. Love it. Any others? Okay, we're moving on. So we had one slide left from Monday's lecture that we need to go through, and it's this question here. Do we really need hypotheses? Do we need hypotheses as we're doing these genetic studies? And you know that the answer is no, right? One of our key terms is hypothesis driven. One of our key terms is hypothesis free. You're like, if we have hypothesis free analyses, then obviously we don't need hypotheses. And then so why do we do this? Why do we do this? And John, you asked an excellent question on Monday. Can a hypothesis-free study spawn or birth a hypothesis-driven? You didn't use those exact words, but I did. And so anyways, a hypothesis actually presents some challenges. First of all, they're going to bias all of your studies towards known variants. Right? If they are hypothesis-driven, you're relying on developing controlled experiments. You can only work with known variants that you already have a suspicion are associated with the genetic illness in some way. Okay? And so you're limiting the scope of your work. It increases the likelihood of a type 1 error. We talked about this with the publication bias, because if you've got all of these different labs and they're all basically asking the same question, you've got all these various labs that are all working on type 2 diabetes. Why? Because people will spend a lot of money to be able to eat whatever they want and not die. Right? So there's a lot of money available in type 2 diabetes research. Okay? Does that make sense? So you have a lot of labs doing work on what are the most important genetic markers and how do they actually work. And so all of these labs are basically targeting the same locations in the genome, doing very similar projects. And if each of them basically have a 20% or a 1 in 5, so that's, no, a 1 in 20, a 5% chance of making a type 1 error, and you've got dozens or hundreds of these, you have an increased likelihood of coming to conclusions that aren't true. When you're saying, yeah, this is how this gene does contribute to this genetic illness, when really, all of those differences were due to chance alone. We're going to play around a little bit with statistics today. Hopefully, we'll have some time. You'll love it. No, I told you on Monday you won't love it, right? Mm-hmm. Yeah, I told you this was the first time that I don't think you will love it, but you'll tolerate it and you'll grow from it.
Why won't we love it?
Oh, you might. You might. Okay. Whole genome screenings do not require hypotheses. And even if you just do partial genome screenings, these don't require hypotheses. Right? You're just basically, you're taking two different groups. You're like, I'm going to take all these DNA samples from people without the genetic illness, and I'm going to take all these DNA samples from people with the genetic illness, and I'm just going to look at pretty much everything, or maybe not everything, but I'll look at a whole bunch of different known single nucleotide polymorphisms. And I'm just looking for any amount of correlation. And so, yeah, we study the genomes of people with the disease. We compare these to genomes of people without the disease. And these studies work really, really well if you can collect families that have at least 2 affected siblings. Okay? Because now you're like, okay, we've got basically both of these siblings have this genetic illness, and we know that they're highly related genetically because they're full siblings. And now we can get into what are the real what are the real loci that are important towards doing this? Because we may have an uninfected sibling or a parent that's uninfected that's very similar genetically, and now we can see fewer differences among those individuals, right? Than if you just took any two random people. Like if I took one random person with the genetic illness, a second random person without the genetic illness, they're not going to be highly related, period, right? But you take two people that are highly related, one with the genetic illness, or a couple with the genetic illness, and a couple without the genetic illness, now you can really minimize the amount of differences that you have to explore as potential contributors to this genetic illness. Okay? That makes sense? It just limits the the amount of variation you have to work with. Because if you take the on average, 54 million SNPs that we have in our genome, right? I've got 54 million, you have on average 54 million. If you take you and I, the amount that you and I share on that, it's going to be fairly low. It's going to be basically whatever the population average is at. But if you take my brother and I, we should share half of those. We should have the same allele at half of those single nucleotide polymorphisms. Yeah?
What if both siblings do have a genetic illness, but it's only expressed under certain methylation patterns or something? How do we know the difference between a genetic illness?
Oh yeah, I mean that's a good question. So if your assumption is that it's a genetic difference and not an epigenetic difference, and it actually is an epigenetic difference, then you're probably not going to find very strong indication statistically that the differences that you see are predicting the occurrence of that disease. So you probably would have to push it to the epigenetic factors. Now the reality is full siblings shared the same womb, and unless their mother's diet was significantly different, their epigenetic signatures are going to be very similar as well. Probably more so than their genetic signatures. So like my brother and I epigenetically are probably more similar to each other than we are genetically. And we're pretty similar genetically. Because it's confirmed we are full siblings. Thank you, 23andMe. Anyways, but we're probably more similar epigenetically than we are genetically. Because we shared the same womb, not at the same time. But the most significant portion of our environment was the same environment.
But it could have been one thing,
oh, sure.
Mom ate carrots one time, and that just.
Yeah, but then if that's the case, if it's only one thing, one difference, that should be really easy to find. If it's only one difference that's influencing that, we would call that a very strong effect. And finding the strong causes are very easy. It's finding those weak causes that's difficult. Right? If it's like half a dozen strong causes that determine, or not determine, but that make you far more likely to develop a genetic illness, we can find those 5 very easily. If it's 5,000 very weak effects that make you more likely to have that genetic illness, that's where we're talking about the challenge. That's where it's difficult. The strong influences, those are easy to find. Yeah.
But how do we, again, if we have full siblings here, let's say genetically they're almost the same, but there's slight differences, we'll assume that the differences are what causes the illness, when really it could be something totally different.
Oh, that's why we would use statistical tools to be able to figure out what amount of variation in the phenotype of the disease is due to these specific differences. I mean, we're not just we're not just analyzing it. I feel like in your mind you're thinking of it as I've got two copies of basically the same book. They're pretty similar, but they're a little bit different. And I'm just going to say well, what is it? What are the differences in this book that actually changes the story, right? We're not just flipping through the book just hoping to find what it is that's the difference. We're doing very robust, very focused statistical tools where we're mapping everything as a potential independent variable against the dependent variable that is the phenotype, right? And so we're finding whatever thing's going to pop up and say, wow, this is having a large effect on either the presence or the severity of that phenotype. Then we've got tools to find them. We're not just hoping to find something good, right? Yeah. Does that make sense? I don't know how good that analogy was. That's just the one that came to me. Kaylin, did you have a question?
How much does it cost, or is it a normal thing for people to get the whole genome screen?
I think it's like $1,500 now, somewhere between $1,500 and $2,000, and it's not very common because it's in some ways it's useless. You'd be better off just screening your exome, right? The portion that's protein coding, even though some of the weak effects are going to be in non-coding genes. But I mean, you'd be better off just sequencing those, or sequencing just a few million of the known single nucleotide polymorphisms, rather than sequencing your entire genome. Yeah, most of the genome, it's the same. It's the same from one individual to another, so it's obviously important, but it's not going to help us to figure out these genetic illnesses. Yeah. But that, of course, I might be wrong, and a lot of scientists were like, why go through the process of sequencing the human genome anyways, because it doesn't matter, it's all about coding genes. Which is obviously more than just coding genes, but it's still not the entire genome. Okay, we've talked about this already, that the probability of making a type 2 error becomes really high when we employ Bonferroni correction, and we take these huge samples, and so we divide our alpha level, which is typically 0.05, where you would have to get differences. The differences between your groups, the probability of those happening by chance would have to be 5% or less in order for us to say that they're significantly different. But if we have 100,000 individuals in our sample, or 1 million individuals in our sample, that's just way too easy to make a mistake. So we'll divide that alpha level, 0.05, by our total number of samples, and then we're like, okay, in order for these things to truly be considered different, the probability of getting this difference due to chance needs to be less than 1/1000%, right? That's what we do with the Bonferroni correction. Well, then what that does is it makes it very easy to make a type 2 error, right? Where you assume that there's no impact of this locus on the genetic illness when there really is. So one of the ways we can get around this in our hypothesis-free studies is to do smaller groups, so your Bonferroni correction isn't as extreme. Maybe groups of 1,000 individuals, and then after the fact, you combine all those data. So in any individual study, you're only having about 1,000 samples, but then you pool several samples worth of data together. And this tries to find a happy medium between minimizing both the probability of a type 1 error, which is basically saying that this locus contributes to the disease when it really doesn't, and minimizing a type 2 error where you say this locus doesn't contribute to the disease when it really does. Okay?
Does it work?
Does it work? Yeah. Yeah. Well, I mean, when biologists don't do the statistics, yes, it works. When you hire statisticians to do this. All right. And then we can use partial genome screenings. I already mentioned this. And this is the world of personal genomics, direct-to-consumer studies. Okay, where they're not sequencing your entire genome, 23andMe, they're looking at about 2 million single nucleotide polymorphisms. They're not even looking at all the single nucleotide polymorphisms, which would be about 54 million. They're only looking at a fraction of them, and then using that to basically compare you to individuals that have these particular genetic illnesses. Okay, they're finding which of those markers correlate significantly with certain genetic illnesses. Yeah.
So, with things like 23andMe,
Yep.
are companies then using that to do their own research? So it's
They
not
do.
just you finding.
Yeah, but you have to give them permission to use it to do their own research. They don't just do it.
Okay.
You have to give them your permission. And I guess somebody about a year ago, maybe it was 2 years ago now, he sent them his data, and they asked his permission to use it for research, and it completely transformed our idea of human origins. Because his Y chromosome was so unique, it basically changed it to where, although you won't hear much press about this, to where the out of Africa hypothesis looks virtually impossible, and it now looks like human origins were right smack dab in the Middle East, which is kind of cool. But anyways, that's at least our species. Our species origins right smack dab in the Middle East.
What do you mean by our species?
Our species, Homo sapiens.
Oh.
Yeah, because I mean, who knows, Homo neanderthals, when they split off from the humankind and where that happened. Probably in Europe somewhere, where they became reproductively isolated from the rest of our kind. So, but not fully reproductively isolated because they still, hybridized with our species.
Yeah. What?
So why call them a separate species?
Yeah.
Morphological species concept, right?
Yeah.
If you can compare it and match it to a type organism, it's a separate species. All right. Any questions about this? Do we need hypotheses? No. And in fact, in some ways, it's nicer not to have them, right? But it's all comparative study, right? So it's all correlations, right? You can't say things like cause and effect. You can't say this mutation causes this genetic illness, like you can with cystic fibrosis. You know, a mutation in the CFTR gene causes cystic fibrosis. Although some people will still argue that. Like, no, it's not really the mutation. It's more the environment or specific pathogens. It's like, all right, well, cool. All right. So how do we determine expression levels? We basically have two big questions that we need to deal with and that the remainder of chapter 10 talk about. First one is how do we determine expression levels? And then the other one is why do we use animal models? Okay, because I already talked a little bit in the key terms, the benefits of animal models. No, I haven't talked about that very much. I more talked about the structure of animal models. Okay, sorry. How do we determine expression levels? First thing we can do, chip sequencing. Chip sequencing, in this case, CHIP stands for chromosome immunoprecipitation. And basically, you're just targeting portions on the genome that are bound by your target protein. So I mentioned this in the key terms, but if you've got a particular transcription factor in mind, and you want to know all of the places in the genome where that transcription factor operates, you design antibodies against that transcription factor, and you let those antibodies do their work. Go and find it and bind to it. And then wherever you find that in the genome, you sequence there. And then you're like, cool, this gene is going to be transcribed by this transcription factor, and so will these other 15 genes or 100 genes. And so now you're like, man, these genes are related in some way. They are epistatic because they are all being transcribed by the same, well, they're not being transcribed by the transcription factor, I know. They're being transcribed by RNA polymerase. But they're all being facilitated by the same transcription factor. And so they are all being expressed at the same time, ideally for the same purpose. Okay? Make sense? So this is one way we can do it. So if we're like, all right, we know that this protein is present in the cell when the cell does this. Okay? Like we know that we find this protein, this transcription factor in the cell when we give the cell this hormone. And we're like, okay, we know this is a transcription factor because we see it go into the nucleus and it has there are certain shapes you look for to tell you that they're transcription factors. And we're like, okay, we know this is a transcription factor, but we don't know all of the genes that it's going to activate. You want to figure that out? You design an antibody against that transcription factor, and you allow that transcription factor to go into the nucleus, then you let those antibodies free. They go and find it. And then you sequence those areas. Yeah?
Wait, you said something is all being facilitated by the same transcription factor?
Yeah, because transcription factors don't transcribe anything. RNA polymerase does.
No, but what is all being facilitated by the same?
Several different genes. Yeah. Anywhere that transcription factor binds at the promoter, it's going to lead to the transcription of that gene. Okay, so that's one. We could also do what's called DNase. Why does this keep autocorrecting? It's supposed to be capital D, capital N, ACE, all lowercase. That's twice now, because I saw it when I was going through and doing my lecture on the key terms. Anyways, DNase, specifically DNase 1, targets very specific regions of the chromosome, and those regions it targets are those that are loosely packed. And if they're loosely packed, it's an indication that they are actively transcribed at some point in the cell's life. Right? Because otherwise, why wouldn't they be fully condensed as heterochromatin? Right? Right. And so this is basically what ENCODE did, is they sequenced the DNA, they did DNA sequencing. Everywhere where DNase 1 cut, they were like, this is a part that's going to be transcribed at some point in the cell's life. But it's almost 80% of the genome. So it's not giving you really accurate ideas of expression, but it's basically giving you an idea of the total expressed amount. Then if you want to know more specific, you can do epigenetic studies. And so epigenetic studies are pretty interesting. We take this molecule called sodium bisulfite. You know, this is sulfite, SO3, SO4 would be sulfate. And then when it's S or H in front of that, it's bi, just like you have sodium bicarbonate. This is sodium bisulfite. And sodium bisulfite goes and converts any cytosine residue that isn't methylated, it converts it into uracil.
You have
I have an error?
Aloe vera.
Oh, who cares? I love aloe. I not to drink it, but it makes my sunburns feel good after I get sunburned. Have you drank it?
Yes.
It's disgusting.
It's really
What's that?
efficient.
Yeah, so is Epsom salt, and I'd rather drink Epsom salt, you know? Like if you just need to get things flowing. Or Metamucil, Miralax, you know? It's pretty gross. Anyways, sorry, that's another story for another time. So the sodium bisulfite, any cytosine that isn't methylated, which those cytosine residues are the main one methylated, right? And specifically a cytosine right before a guanine, right? You remember that? The CPG, those are target methylation sites. Okay, so any cytosine that isn't methylated, it'll get converted to uracil, and then you sequence it. And so where you find a uracil, you're like, that's weird, there should be uracil in DNA, but you're like, I put something in that converted cytosine into uracil, but that means that cytosine wasn't methylated. So any cytosine that stays cytosine when you sequence it means it was methylated. And so that portion of the genome has an off switch. And if it has an off switch, it means it's not being expressed, unless, of course, it's an off switch that's being ignored. Because it's an off switch in an area where the histones are acetylated. Right? Yeah. Because the methylation at the level of the genome, it's the off switch that can be ignored. That was on your midterm. Yeah. Anyways, I have those to give back to you, too.
Grade them already?
Yeah, I did grade them. I have not put them in on Canvas, but I'll give them to you. I scanned them so I can put them into Canvas. I don't know what the average on the multiple choice before correction.
Correction.
Well, before I corrected it to make the average higher. So the average beforehand, it was not good. It was like a 65. I think was the average on the Scantron portion. The average on the short answer portion was like just below an 80, I think. Like right about there. And then so I corrected the multiple choice to make it to where the average was now pretty comparable with the short answer. So overall the average was somewhere right around 80. Maybe a little bit lower, 78, 79. I'll see when I'll put them into Canvas, then they'll tell me what the overall average is. Yes, one person did get 100%. But only one. No, nobody got 100%. Wait. No, somebody got a 199 out of 200. Initially, I had it as a 200, and then I was like, wait, my math was wrong, and I went back and I scribbled it out and replaced it with
Was that
199.
including only kind of 4 highest essays?
Yes, I did that for everyone. Yeah. So for everybody, I just took your 4 highest scores on the essays, and then used those scores. Okay, anyways, it's not time to talk about the midterm yet.
Can you summarize what you just said about this point? I am good with that.
Oh, yeah. So with epigenetic studies, if you're wanting to look whether or not a section is methylated and therefore shut off, what you do is you use sodium bisulfite, which will go in, and anywhere where you have an unmethylated cytosine, it'll convert it into a uracil. So then when you go and sequence it, if you find a uracil, you know that that was originally a cytosine and that it was not methylated. But then if you sequence it and it still says cytosine, you know that that cytosine is methylated because it wasn't converted into uracil.
Okay.
So then it tells you where where your DNA methylation is. Yeah. Then you'd also have to study the histones to know for sure that that off switch is really an off switch, right? I mean, it's always an off switch, but you know if it's an off switch that can't be ignored. Yeah.
Is there like a basic mechanism of what does it use, like a special polymer?
How it converts cytosine into uracil?
Yeah.
Cytosine and uracil are very similar.
Yeah.
Cytosine, thymine, and uracil are all very similar chemically. So as far as what it's doing, you'd have to ask an organic chemist. Yeah.
So it's actually changing the cytosine, it's not replacing it.
Correct. No, it's not replacing it with the uracil. It's chemically modifying the cytosine residue into uracil.
No,
Yep. All right, real-time PCR. Real-time PCR is used to determine relative concentration. And so real-time PCR works in a lot of ways like actual PCR, where you design a specific primer for a specific region, for a specific nucleic acid or region of the DNA, and then you supply DNA polymerase and you supply nucleotides and you let DNA polymerase do what DNA polymerase does best. And that's write new nucleotides, but it can only do it when there's a primer in place, because DNA polymerase is not self-priming, right? And that's why when the DNA is duplicated, you have another enzyme called primase that comes in, writes an RNA primer, and then DNA polymerase takes over. Yes? You remember that? Okay. So that's how PCR works. But now we can design primers that will only target a specific RNA, a specific type of mRNA. And we design a primer that'll target that. And so if that mRNA is present, that is, if that gene is being expressed, your primer will have something to bind, and you'll start making copies of that RNA by the millions, and then eventually, because you used only fluorescent nucleotides, eventually you have enough of it that the computer sees the glow. And you're like, cool, that RNA was present. And now, however many cycles you have to go through before it becomes present tells you how much there was in the beginning. If you had a lot in the beginning, you shouldn't have to go through as many cycles before the computer recognizes its presence, right? If you had very little in the beginning, you have to go through more cycles before you get to a high enough concentration that the computer recognizes its presence.
Okay, wait, sorry. So if it binds to that specific section of DNA, that means what?
I mean, it doesn't mean anything. I mean, you design your primer to bind to a specific region. But if your primer that you designed for a specific mRNA actually works and you get polymerization, it tells you that that mRNA was present. Because otherwise, there would be nothing for that primer to bind to. Right? Right. So this is real-time PCR. It's cool, but all it does is really give you relative concentration of RNAs. So it can tell you, is that gene being expressed, yes or no, and approximately how much. Okay? But no absolute values. But here's where microarrays come in. Microarrays, we've already talked a little bit about these. These can give you a little bit better idea of a quantitative amount of the RNA that's being produced. Because if you've got several of the same probes on the same chip, right? You've got this chip, and let's say you have 500 of the same probe. And all of them bind to your target RNA. Now you're like, we've got a lot of that RNA, right? But if only half of them bind to that target RNA, now you're like, we have less than a lot. I mean, it might still be a lot. But you can come up with, it's a little bit easier to quantify approximately how much you have, and to not just say relative. Like this, I had more of this here, I had less of it here. You can come up with a better way to quantify it. Now, what you can't do with microarrays that you can do with RNA sequencing. So RNA sequencing, you can determine the presence of an RNA, the concentration. We can do both those with a microarray, but what we can't do is determine how it's been modified. How it's been modified. And so we can do this with RNA sequencing, because you have to think about this. If you've got a probe, and your probe matches 20 out of the 30 nucleotides, guess what's going to happen? The probe's going to attach, right? If your probe matches 20 out of the 30 nucleotides, you're going to form, just statistically, you'll form 2.5 hydrogen bonds per attachment, right? Because ATs have 2 hydrogen bonds, GCs have 3 hydrogen bonds. So on average, if the number of G's and C's are the same to the number of A's and T's, you'll make 2.5 hydrogen bonds for every attachment. Does that make sense? So if you attach at 20 out of the 30 places, that's 50 hydrogen bonds. If you attach at 30 out of the 30, that's, 45? No. Wait, no. 2.5 times 30 is 95. No? No, 75. There we go. It's Wednesday, my math should be better. But you have 75 attachments if you get all 30. You have 50 attachments if you only get 20 out of the 30. But guess what? You still have 50 attachments. It's still going to bind. So it doesn't really tell you if that RNA's been modified. But if you sequence that RNA, you know the exact sequence. You know how it's been modified, how it's been alternatively spliced, what's remaining as an intron. Does that make sense? Okay. So this gives you presence, concentration, which microarrays will do as well, but it also tells us how this RNA has been modified. Real-time PCR will give you presence and relative concentration, not absolute.
Wait. So this last one does
All 3?
absolute.
Yeah, but it's labor intensive and it's kind of expensive, not because of the materials. Microarrays materials are a lot more expensive than RNA sequencing, but man hours. There are a lot of man hours whenever you're doing any sequencing work.
So microarrays gives you relative.
Or woman hours, sorry. I meant it generally.
People hours.
Yeah, people hours.
So microarrays gives you general concentration, I mean relative concentration, and then the last one,
It gives us absolute.
absolute.
Yeah. So microarrays give us absolute as well,
Wait,
but not how they've been modified. And then the last one I want to talk about, this one, it just has a fun name, chip on chip. And so this is where we use a microarray combined with chromosome immunoprecipitation. So we in order for something to bind to your chip, not only does its sequence need to match what your target sequence is, but the antibody on your chip also has to match the protein that comes with it. Does that make sense? So you're not just targeting a specific region of the genome, you're also targeting that transcription factor. So you're looking at, is that transcription factor present? Yes. Is the RNA product also present? Yes. And so now you're like double yes. The right transcription factor. So it basically gives you up to the minute, is this being transcribed right at this moment? Right? Because what if the RNA had been transcribed, but it stopped transcribing it already? So that gene was expressed, but it's no longer being expressed, but those RNAs haven't been gobbled up by nucleases yet. And then you're like, oh, this is a positive result. This gene is being expressed. And you're like, no, it's actually not being expressed. It's on its way out. Like nucleases are chewing it up. The cell stopped expressing it. But if the transcription factor is still present and the RNA is present, now you're like, this was being expressed at the very moment that I killed this cell. Yeah.
And that combines microarray and what else?
Oh, and chromosome immunoprecipitation. We didn't get through all of our material, and I need to hand you your exams back. We only have like 5 minutes. So, actually we have 7. But we're not going to get through all of our material, nor are we going to get to our statistics, which means that you are not going to be prepared to take the quiz. No, it's not fine. What I will do is, the quiz is going to open as soon as class ends. If you want to go and take it, feel free to go and take it. I'm going to encourage you not to. And I will just delay the due date of the quiz until Monday. And then that way you can wait until after our class together on Friday to prepare. Okay? All right.